Hello students, good afternoon. Now in the last lecture we have completed in the living world till the taxonomical hierarchies. Okay, today I will take you through the last topic in this chapter, that is the taxonomical aids. Okay, what are the taxonomical aids? Let's discuss about taxonomical aids. Taxonomical aids. Okay beta. So when we say taxonomical aids means the tools. Taxonomical aid means these are the tools. Okay, the different tools. What are the different tools we are using in the taxonomy which helps in the taxonomical studies? They are known as the taxonomical aids. Okay, so those tools, there are different tools, there are different aids which helps us to get the taxonomical studies in a convenient way. Okay, to understand the taxonomy in a very good way. Okay, so first tool or the first aid in the taxonomical aid is herbarium. It is the herbarium. Okay, so what is the herbarium? Herbarium is a tool. Herbarium is a tool. It's a tool or aid, taxonomical aid, which is prepared by preserved, dried and pressed plant specimens which are stored in a particular place. ठीक है ना? So this is the tool herbarium. Herbarium एक ऐसा tool होता है बेटा जहां पे प्लांट स्पेसिमेंस को प्रिपेयर किया जाता है, ड्राई स्पेसिमेंस, ठीक है, और प्रेस्ड स्पेसिमेंस जो है उनको बाद में किसी अच्छी जगह पे, प्लेस पे प्रिजर्व करके रखते हैं, और उसी प्रिजर्व्ड स्पेसिमेन को हम जब किसी जगह पे स्टोर कर लेते हैं, किसी जगह पे स्टोर करके रखते हैं, तो उनको प्रॉपर तरीके से स्टोर करके रखते हैं। उस स्टोरेज हाउस को जिसमें ये प्रिजर्व्ड स्पेसिमेन ऑफ प्लांट स्टोर करके रखते हैं, दैट particular storage house is known as the herbarium house. Okay, so this is known as the herbarium. That is a tool. Now this tool actually is going to be prepared by the different methods. There is a different methodology how to prepare the herbarium. See, if we go to the methodology, of herbarium, how to prepare this herbarium. There is a proper strategy, there is a proper methodology for the preparation of the herbarium. Number one, like first there is the important thing, that is the sample collection. For the herbarium, to prepare the specimen of the plant, especially the specimen of which plant is going to be selected. The plants who are going to be endangered, the plants who are going to be nearby extinct, those plants, they are being preserved, their morphological characters are being preserved in a particular place or in a particular store. So such plants are being chosen, and for those plants the sample is being collected from those plants. Now this step is a very crucial step. Under the preparation of the herbarium, this step is the very crucial step, because for the sample collection Nobody know, these taxonomists, they know that this species which is going to be endangered or which is going to be nearby extinct, that might be in somewhere in the Himalayan jungles, that might be in Amazon jungles. Okay, so any researcher or any collector has to visit to those places which are very risky. There are the many number of risky animals are there. There is a risk of the life as well. Okay, so taxonomists or the collectors, they find it difficult to visit to those places. So that is why sample collection is it's a very difficult task. Okay, it's very crucial step. So now the sample, for the collection of the sample, there is also some guidelines and rules has to be followed, like the sample size. The sample should be a healthy branch of the plant, that plant whose specimen we have to prepare. So first, we cannot take whole plant as a specimen, we cannot take any part of the plant as a specimen. We have to choose a specific part that should be a healthy branch of that plant, that branch which should depict its leaves. Okay, there should be a presence of fruits, okay, as well, which can depict its morphological characteristics of the plant. Okay, even seeds should be also there. So such kind of the branch of the plant is going to be chosen for this specimen. So once this sample is going to be collected, so second step is drying of sample, drying of the sample. Okay, once the sample is being collected by the researcher or the taxonomist, that sample is properly taken care till it reaches to the lab or till it reaches to the office somewhere. Now in the lab, this sample, it is inside, having the presence of starch, having the presence of water. Okay, so it is going to be dried. It is going to be dried by using papers, by using some paper. So papers, under the paper, wrapping up the paper, these specimens they are going to be kept inside that. Okay, and after 1 or 2 days they are going to be dried. So this water source and starch source, it is going to be removed from them. So second step is the drying of the sample. This sample is going to be dried. Third, this sample is going to be poisoning. It is going to be poisoning. Poisoning means here, this sample might be having the chances that it may get contaminated, okay, by microbes or by fungi. So for that purpose, there is going to be prepared a solution of HgCl2, mercury chloride. This solution, 0.5% molar solution of this mercury chloride is being prepared, and it is going to be sprayed. It is going to be sprayed on the sample. Okay, so this solution is sprayed on the sample, which prevents it from the contamination from the bacteria as well as the fungi. Okay, but while doing this spraying with the HgCl2, again the sample attains the moisture. Sample attains the moisture. It attains moisture. So again it becomes wet. So there is again another step is required to make it dry, and again drying is done. Drying is required. Drying is again required. So same, the second step is again going to be followed here. Drying, again these samples are being dried. Now once it is dried, just a moment, the board is going to be loading. Okay, so sorry for the disruption. So fourth step was the drying. Now next step, fifth step, after the drying, this sample, it is going to be, this sample is arranged in proper size, in a proper size. Okay, so the whole sample cannot be depicted on the paper. Okay, so that there is a proper size. This sample should be in the proper size according to the international size. Okay, so for that purpose, then a cardboard, special cardboards are prepared. Cardboards are prepared in size, in a proper size. The standard size for this is 11.5 into 16.5 inches. 11.5 and into 16.5 inches. This is the international size. This is the international standard. International standard size for herbarium, to prepare the herbarium specimen. Okay, so इतने size में जो है, on an average, this cardboard has to be cut like this. Okay, so this is the 11.5 and this will be the 16.5 inches. Okay, like this. So in this manner, the cardboard is going to be prepared. A specific type of the cardboards, they are known as herbarium cardboards. These are known as specifically herbarium cardboard. So that cardboard is cut in the proper size, and on this cardboard, this sample is properly cut, and next is mounting. Next is mounting. Okay, so this sample size, suppose this is the sample of this cut part stem of that plant, which depicts the root system, then shoot system. Okay, then maybe flower may be present over there, it may have the fruit as well. Okay, so all these things, they are being cut in a proper size, then it is going to be pasted. That is called the mounting. Okay, so properly mounted in this proper size of the cardboard. Okay, then again after the mounting, it is going to be stitched. Okay, stitching is being done. Okay, so small stitches are being here adjusted, that properly aligns the sample on this cardboard. Okay, now once this is done, then at the below corner, right side corner, some space is being utilized here. This much space is being used, and in this space, particular, this space is being utilized for, in this space there is going to be written a collection date of the sample, on which date this sample of the species is going to be taken. At the date, proper date is going to be mentioned. Then collection place, at which place this sample has been collected. Then collector's name, who has collected this sample. Okay, that whose name it is going also depicted. Then plant, its plant's vernacular name. Vernacular name means local name. Okay, what is its species local name over there, from where it has been collected. Then its English name, what is its English name. then its scientific name. What is its scientific name? That is going to be written. Then what is the genus? What is its genus? And what is its family? From which family it belongs to? Okay. So this type of information is being written at the below lower right corner over there. Okay. And after that, this whole cardboard is going to be sealed. It is going to be sealed with a glass, ट्रांसपेरेंट ग्लास. ओके, तो आउटसाइड से जो है इसको पूरा ट्रांसपेरेंट ग्लास से कवर किया जाता है. ओके, देन इट इज गोइंग कंप्लीटली रैपिड around that, no oxygen should go inside. Before that also remember, before wrapping the whole transparent glass, it is also being sprayed with the again poisoning, because it is going to be now sealed completely for maybe a longer period of time, so it should not get contaminated. For that purpose again it is being before poisoning for that. So after that, this, when it is going to be prepared, a particular sample with its information representing here, this is known as a herbarium. This is known as the specimen, proper specimen of that plant species. Okay, now this specimen is going to be stored. It is going to be stored. It is going to be stored in the house or a place wherever the government has chosen to take these specimens, to store these specimens. That place, that storage is known as herbarium. That is known as herbarium place. Okay, so everywhere in the country there are the different herbariums, there are the different herbarium centers. Okay, so those herbarium centers, they contain the thousands and millions of these kind of the specimens inside. So this kind of tool, this kind of the taxonomical tool like the herbarium, how are they helpful to us? How are they helpful to the society? Now remember, this herbarium, the herbarium place or the storage, house storage where these specimens are being stored in a proper way, that functions as a quick, it functions as a quick referral system. Quick referral system. So whenever any researcher, whenever any scientist, any researcher wants to do the research on the same species, then this specimen becomes the quick source of the information. Quick source of information from that researcher can visit to the herbarium and get the basic information about that species. So it functions as the quick referral system. Okay? So remember, there are some famous herbariums throughout the world, which are very famous herbarium, like Kew, in Kew, England, there is a national herbarium. In Kew, England, there is a national herbarium center which contains on an average 6.5 million specimens. 6.5 million specimens are preserved over there. Okay, so similarly in India also there are the various herbarium centers where the different specimens are being preserved. Okay, like there is the Indian Herbarium Center, okay, which is located in Kolkata. It also contains 3 to 3.5 million specimens. Okay, so almost in every state there is a small herbarium center, there is a small place where such kind of specimens are being preserved. Okay, then any researcher, taxonomist can visit to that herbarium center and get the quick information, taxonomical information about this specimen. This is how the herbarium is being prepared and it becomes the quick referral source. Okay, remember this is the first taxonomical tool. Similarly, there are the other taxonomical tools like botanical gardens. Botanical garden. Okay, beta. So there are the other tools as well, botanical gardens. Now, what are the botanical gardens? Why the government is preparing, why the government is forming and developing these botanical gardens? What is the requirement of that? Botanical gardens are being developed only because there are some species which are nearby to extinct, or endangered species in plants. So there are some species which are very nearby to endangered or going to be extinct in the plants. Such species are being taken care. Those species are being taken from the different areas, and are being taken care or being developed in a particular land. That particular part of the land is being chosen, okay, which is under the human control. Suppose this is the particular land, part of the land, and this land is properly managed by humans. Okay, and in this land, these endangered species of the plants who have been collected from the different geographical areas, and those from different geographical areas, they are being completely grown in this tract of the land, okay. So those plants which are being grown in this part of the land are being properly taken care by the humans, okay, and they grow well. Even their production is being increased. So such species which are going to be endangered or extinct, they are being preserved, they are being conserved, okay. So such species are being protected in this tract of the land. So this kind of tool, it is actually what it is known as ex situ conservation. It is known as ex situ conservation. So it means the different plant species endangered or going to extinct are being taken from the different geographical areas and being developed in a proper tract of the land under the human control, are being saved, are being prevented from the endangering. So that is called ex situ conservation. So this is the ex situ conservation of plants or plant species. So that is the main objective behind to form the botanical gardens, So in the botanical garden you will find these different types of species of plants are being grown over there, they are being reproduced over there, okay. That is the main objective behind this, why the government is forming these kind of the botanical gardens. Apart from that, these botanical gardens, they also provide us the aesthetic values. They also provide the aesthetic values to the society. So we are visiting to these botanical gardens, then we feel very happy over there because the natural environment is, natural beauty is there. We can see the flowers, we can see the seeds, we can see the branches of the plants. Even when we visit to the botanical garden, everyone who visits in the botanical garden, on this plant stem there is a small plate like structure is being pasted on the plant, and on that plate there is given a written information about this species, okay, about its genus, about its family, okay, about its scientific name, and basic information is written on those plates. So one who visits to the botanical gardens can easily get the basic information, basic taxonomical information by that. So that can easily get the overview, clear view about that plant, how it looks, how is it branches, how is it leaves, how is it flower, seed, etc, etc. Okay, so that is the also helpful for taxonomical study. So that is why botanical garden is also the another tool. Okay, remember this. Next tool in the same way, botanical garden we have is the zoological parks. Zoological parks. Okay, beta. So zoological parks, like the botanical gardens, why the government is developing the zoological parks? There is also the logic behind this, that there are endangered or nearly extinct animal species which are being conserved in a particular tract of land. So it's the same way, like we conserve the plant species, in the same way there are the conservation of the animal species. Those animals who are vulnerable, who are endangered, who are very nearby to the extinct, okay, such animals are being taken from the different geographical areas, okay, and those animals are being kept in a particular land which is completely under the control of the humans, managed by the humans. And those animals, suppose this is the tract of the land chosen for these animals who have been taken from different geographical areas. So in this tract of the land, this land is completely, you can say, fenced outside, okay, it's completely being prevented that any animal should not go out of this boundary, okay. Second thing, these animals in this tract of the land, they should feel that we are in the natural environment like they were in the earlier environment, okay. So they should feel like that we are in the natural, otherwise they cannot stay over there. They will always try to move out from this place. So for that purpose, there also the plants are being grown. Okay, plants are being grown. Similarly, those plants again who are endangered, or maybe some other plants who are not endangered, mixed way, they are all going to be grown in this tract of the land, so that these animals, they should not feel that we are in any kind of a desert kind of a thing. They will not stay over there. So then animals, they feel we are in the natural environment and they stay over there, and the proper feeding purpose, the arrangement is being done for their feeding as well, okay. So these animals are being conserved, they are also being protected from being endangered or extinction. So this is also, zoological park is also meant for, there is objective, it is also called ex situ conservation. Ex situ conservation of animal species, okay. So from the different tracts of the geographical areas, these species are being conserved, okay. That is the main objective behind that. Also, when we visit to these zoological parks, They are known as now zoological parks. When anyone visits to the zoological park, also get the chance to know about the behavior of the animals. Behavior of animals, okay, their feeding, how they are feeding themselves, their behavior, how they react to the things. So that kind of animal behavior we are going to understand by that. Even when we visit to the zoological park, every cage outside of that animal, there is written the some information that gives a basic information about that animal. That is also the taxonomical tool. So this is the another taxonomical tool going to be used. Next, there are the another taxonomical tools. Okay, there are many taxonomical tools. Like another taxonomical tool is called museum. Okay, now you all must be knowing what is the museum. Okay, museum is meant for what? Museum is also meant for preservation of, there are the preservation of living as well as dead specimens of plants and animals. प्लांट्स एंड एनिमल्स, दोनों प्लांट एंड एनिमल स्पेसिमेंस को जो है म्यूजियम में हम दिखाते हैं ना. मोस्टली लिविंग थिंग्स बहुत कम होती हैं, बट मोस्ट ऑफ दी स्पेसिमेंस वहां पे डेड नेचर में होते हैं, डेड फॉर्म में होते हैं. सो दोज प्लांट एंड एनिमल, दे कुड बी इधर प्लांट, दे कुड बी इधर एनिमल्स. ओके, सो इन द म्यूजियम्स इफ यू सी द प्लांट्स as well as the animal specimens are preserved in a solution called formalin. There are small jars in the museums where those specimens are kept in the jars, but in that jar there is the solution. That solution is known as the formalin, and inside the formalin they remains in the smooth condition. Okay, so these specimens are being preserved. Also in the museums, the large samples, large plant and animal specimens are being kept in fragmentation, or being kept in a fragmented manner. If any specimen, if any animal is larger in the size, okay, that animal is not being kept in a same way, then it is going to be stuffed. Its body is going to be stuffed into small fragments, like elephant. So the whole elephant cannot be depicted in the museum. Its head part is going to be cut and kept preserved somewhere in the museum. Legs being cut, arms being cut. So it's a stuffed parts of the body. They are going to be depicted over there, okay, in the museum. So not in the whole animal or whole plant species. So museums are being like that. Okay, so both plant and animal specimens, especially in the dead condition, are being kept in the museum. So whenever you visit to the museum, so you get the basic taxonomical information about these animal and plant specimens. Okay, so they are also functioning as a taxonomical tool. Okay, similarly there are the another tools. They are known as like flora manuals, monographs, catalogs, okay, and keys. These are the another type of the taxonomical tools. Now I will take you through this. Like flora, when we say the flora, you can imagine that there is a book, it's a larger book, and this book contains, it gives the information about the distribution and habitat of number of plant species in a particular geographical area. So this is a kind of a book which gives the information about that there is a particular tract of the land, there is a particular geographical area, that which plant is present in that area, in which habitat this plant is growing well, and what kind of the root system, shoot system is there in this plant, fruit, seeds and everything, in which habitat this plant is available, where it is distributed in that geographical area. This whole information, if some book is being prepared like that and gives us this kind of the whole information, that is known as flora. So for example, when we say flora of Delhi, so what does it mean then? Then it depicts here what is the total number of the plant species, different species of plants available in the geographical area like Delhi. Okay, so it gives the whole habitat and distribution of the plant of that area. So that is known as the flora. So that is also remaining as a specific taxonomical tool. In the same manner, you think about that there is another book. Imagine there is another book which is like the flora, but it does not give us the complete information of all that information which the flora is giving. Okay, so this is the book which gives only information about the distribution of plants, plant species in an area, in a particular area. So it is giving the information about distribution, where these different types of plants are available and growing in that particular area. A little bit less information than the flora, that is known as the manual. So it also serves as a taxonomical tool. In the same manner, monograph. Monograph is the small booklet. It's a small booklet type which gives the information about one taxon, or category. So like I have taken you through already the taxonomical hierarchy. So there are 7 obligate taxonomical hierarchy. So if any book is there and that book gives the information out of the 7 categories, it gives the information about one category, like family. So which family plants are available in the particular area. So it's a short information. Okay, so that kind of the book is known as the monograph. Remember this. So monograph gives us the information about the particular one taxon or category. Now catalog. So imagine the catalog is also a very small booklet. It's very small booklet which gives the information of plants, which plants are available in an area in alphabetical order, Okay, so this is a small booklet. Imagine that small booklet which is giving the information in the alphabetical order, like first it will give us the plants who are starting with the A, then B, then C, then D, like that. Okay, in the alphabetical order. So there are the few number of the plants which are available in that particular area, and they are arranged in the alphabetical order. Okay, so the small little information type of the booklet, it is known as catalog. So it also gives a minimum information. Okay, now another tool is the keys. It's called a key tool. Key is a tool which is a modern tool. It is the modern tool, and this tool, it is analytical tool. It is the analytical tool. It is based on the analytical analysis, like it gives the analytical situations. This tool has been designed by the taxonomists to make their classification convenient. So for this tool key, key tool, if we talk about the key, it's an analytical tool, and the taxonomists, they choose the different criteria, they choose the different features, like that they choose the character for each species or each category. Out of the 7 category, every category they choose for every category they choose different types of characteristics, okay, or different types of feature, and those characters they choose in this, they choose the contrasting characters, and based on these contrasting characters which are analytical in nature, they choose to classify the species. Like for example, a contrasting character, like there is a character known as height. There are suppose the taxonomists, they wanted to differentiate between the species A and species B. So between the two species, they choose the feature, they choose the character height. Based on the height, they will see which species is having the more height, which is having the less height. So this height is a character, but this height may have two options. It may have two options, like there could be the tall and there could be a dwarf. So tall and dwarf, these are the two options of the height. There are the two options of the height character. Okay, so when the researcher, when the taxonomist has to differentiate between species 1 and species 2, and he has taken the criteria is the character height. Okay, so in the height he will see only whether the species 1 and 2, who is tall and who is dwarf. So he has to choose whether this species is a tall and then another will be the dwarf. So it is a situation like giving the character in a yes and no form, like yes or no. So the researcher or taxonomist, he has to choose only one out of these two, either yes or either no. Okay, either black, either white. Okay, either red, either white, either tall, either dwarf. So such kind of statements, such kind of contrasting character, this contrasting character, it is known as couplet. It is known as couplet in the taxonomical language, in the taxonomical nomenclature system. Okay, this character, contrasting character, it is known as a couplet. Okay, but if you see inside the couplet, finally a researcher, a taxonomist, finally which statement he choose to differentiate between the two species or more than two species, that is the statement either tall or dwarf under the character. So these two are two different statements. So these are statements, statement 1, statement 2. This is the 2 statement. So these statements, they are known as leads. They are known as leads. In the taxonomical nomenclature, they are known as leads, and the contrasting character, it is known as couplet. So the couplet, and finally we choose the lead. It is the lead which helps the researcher and taxonomist to differentiate between their species. What is the difference between the species? Okay, so that is how, by this system, this advanced system has been developed by using computers. So inside the computer, such characters are being designed for the each character, contrasting character, and every character has a two statement, they are called leads. Then the computer, they are giving the command to the computer that there are the 100 species. Okay, then computer is giving the command based on the height, 100, more than 100 species are differentiated at a time. Okay, so they give a classification system more conveniency. Okay, that character, that type of this new tool which helps in the taxonomical studies to get more convenient is known as the key. It is known as the key. ठीक है ना, ये याद रखो. ठीक है बेटा. So these are the important taxonomical tools of this chapter which helps to understand well the taxonomical studies. Okay, I hope you have understood about this taxonomical tool topic. With this we are complete, we have complete this chapter. Okay, thank you very much. Next we will deal with the next chapter. Thank you.